eat in the class, it's kind of hard to get lunch and be back on time. I'm sorry for that. It's just university regulation. We have to keep masks on at all times, so we can't eat in class. Okay, so we can get started where we left off last time. Let's see. Sharing this screen. And it's not liking. All right, I have to exit. Sorry, I got you to be quiet to listen to me, and then now I have technical things. Okay, here we go. Let's try this again. Okay, good. Now it's working. All right. So we just barely started this lecture last time, but we talked a little bit about sample plan or sampling plan. So what is a sampling plan? Can you remember from last time? What does the plan entail? Go ahead, Zoe.
It's like a plan of how you're going to choose which samples to take from the lot. So if it's going to be random or how many, and then how you're going to compare them, store them, all that kind of stuff.
All that kind of stuff. Yes. Yes. So deciding on how many samples, how are we going to select them from the lot, how we're going to prepare our sample, transport our sample if we need to, and store our sample. So it's kind of the entire process of getting a sample and testing up until testing. So it does not include testing, it includes everything that goes on before we start the assay. So why is sampling plan so important, Drew?
Because you need to pick a sample that will accurately represent the lot. If you pick like maybe the first scan level lot, that might not be the representative of what may get scanned in the middle or first scan.
Absolutely. So it's very important for selecting, knowing if it's going to be, we need to select 12 samples for nutrition labeling based on FDA. So are we getting 12 samples? Are we getting it from which location? And is it a homogeneous sample or a heterogeneous sample that needs homogenization? How are we going to prepare our samples, store them, etc.? Any mistake anywhere along the line is going to impact big deal the decision. And the decision can either harm the consumer if it's a safety situation, or it can harm the vendor, like we talked last time, money. Are we going to reject the lot when it's not supposed to be rejected? Then we're going to cost the vendor money. So it's very important in terms of getting a representative and accurate determination that would impact the decision and also be, protect the consumer and protect the vendor. Okay, so the others are great because we haven't gotten to that yet. So these would be questions you would ask yourself later after you study. Okay, so the factors that affect the choice of sampling. This is our sampling plan. There are a lot of factors, and this is table 5.1. This is good, informative. I don't expect you to memorize all of this. It's just good FYI. So how to think about sampling plan. All right, so first, what's the purpose of the inspection? Is it to accept a lot, to reject a lot? Is it for general quality assessment? Is it for nutrition labeling? So you have to ask yourself the purpose, and based on that, there might be regulations. If it's for nutrition labeling, we have to follow FDA and FSIS regulation for number of samples to withdraw. So nature of the product, you ask yourself, is it homogeneous? Is it milk? So I'm going to take a sample from a bottle of milk. Doesn't matter where I'm getting my sample, it's a homogeneous sample, or is it a heterogeneous sample that I want to make sure, for example, maybe first I need to mill it to reduce sample size before taking the sample out. So thinking about that is important. What is the unit size? Is it the unit size? Is it sacks of flour, like 100 pounds of flour, or is it cans? Like I have boxes with 50 cans in each box. So it really depends on what is the unit size in determining how you're going to take out samples. There are a lot. I'm not going to walk through all of them, but these are examples. Nature of the test method. Is it a destructive test or is it a non-destructive test? So am I doing metal detection? That's non-destructive. Am I doing FTIR or NIR? It's non-destructive. Or am I taking a sample to use it for, let's say, protein determination with nitrogen analyzing unit? That's a destructive method. So what's the nature of the test? Determine how I'm going to use those samples for testing. Is the lot large but uniform, not consistent or small, easily identifiable subunit? It goes on and on. So there are a lot of questions you would ask yourself while you are preparing your sampling plan. Again, I don't expect you to memorize any of this, but when you are working in industry and you have to determine a sampling plan, you want to think about these questions. You have the book as a resource to take out and look at that. If you ask me, am I going to bring a question in the test to tell me in what situation you would choose a sampling plan, I won't go that specific because I'm not going to have you memorize this. Okay, just wanted you to know. All right. Attribute sampling. So there is a variable sampling and there's attribute sampling. When I talk about attribute sampling, it would be a certain characteristic that is not normally distributed in your sample. So let's say Clostridium botulinum, or let's say aflatoxin. So it's really, you might have an organism growing in some regions and secreting a toxin that is not uniformly distributed. So in this particular case, you have to do a lot of sampling. So you have to take a lot of different samples because you want to make sure you covered your basics. Because if it's located in certain region and you take from another region, you might risk the safety of the population by saying, oh, I don't find Clostridium botulinum, I don't find aflatoxin. So we're good to go, but then they are toxic and that's risking the consumer safety. So in this case, it's large sample size. You may make sure you take samples on many locations and have multiple samples to test. So it's usually checking for presence or absence and then decide acceptability. So the example here, Clostridium botulinum in canned food, you should have 0 count. Aflatoxins, some aflatoxins are very, very toxic. You should have 0 count. Some others, you can have a certain number, amount or less to be acceptable. So usually if you have an organism of concern or a toxin of concern, so it's either present or absent. If it's present or absent type of test, so you would have accept. If you want to accept this organism in food, it has to be a certain count and less than particular count. If you shouldn't have anything in there, count 0, then you would reject directly. If you have more than 0, if you have 1, you reject. Variable sampling, these are for constituents that are normally distributed among fat, ash, moisture, everything. So the attribute, it depends on what you're testing. Attribute, what are you testing for? Are you testing for the presence of Clostridium? Are you testing for the presence of aflatoxin? So something that is unique and not usually present across the sample even. distributed. Aflatoxin is produced by a fungal type of fungi. Okay, so it might be growing in a certain region and not all over the food. Okay. Yeah, so they might be located in places that are random, not normally distributed, not evenly homogenized. Yeah. In this case, variable sampling is for those that are normally distributed, like I said, constituents, calcium, protein, moisture, they're normally distributed in your sample. So in this case, you have smaller sample size because you don't need, they're not randomly distributed, they're normally distributed, so you don't take out a lot of samples to analyze. The risk here is low. So, and it's a quantitative measure, it's not qualitative, present or not, or above a certain number or below a certain number. Here you actually measure the quantity that you have on a continuous scale, and then decide acceptability based on what's are your standard of identity, for example, measures. So if a product needs to have a certain amount of fat and you have less than that or more than that, then you decide acceptability. Okay, so quality or specifications that you are after. Then compared to acceptable value. Here's just example of those. So the selection of sample size depends on the expected quality and sampling cost. These are major considerations, but also we need to take into account that sometimes we have regulations to follow. FDA and FSIS. If we look at nutrition labeling for FDA, it says a minimum of 12 samples from a lot. So, and then that's composite sampling. So what does it mean? So you take 12 samples and you compose them together. You bulk them. And then from there, you take 6 subsamples for testing. That is FDA regulation. You take 12 samples randomly, and then you combine them, that's composite sampling. And once they're homogeneous, you take 6 subsamples for testing. FSIS says 6 samples, not 12. Okay, so that's the difference, but that is something that you have to take into account when it's a federal regulation. Drew, just to say, yes, you have a question?
Yeah, so you said like FDA is 12, and then you said the FSIS like 3 or 6.
Oh, both require subset of at least 6. So once you, the FSIS, remember this regulates poultry and meat and eggs, I believe, but FDA everything else. Each one of them tells you how much you have to withdraw and make a composite. Once you have a composite, you need 6 subsamples, like your sub-replication, basically for your testing. See?
But is that just their minimum for all food products, or do they have specifications based on certain food products?
No, that's a minimum for all products if you're doing, for example, nutrition labeling. But we're going to talk about how do we get those samples.
Or do they do more than that?
So if it's for nutrition labeling, they try to do the minimum because it has cost associated with it. Okay? If it's not nutrition labeling and it has to do with safety, you have to do how much you need to do to make sure that the product is safe. Okay. So if we're doing acceptance sampling, there are different types. You have the single accept reject based on single sampling, and here's what it means. Single sampling does not mean you take one sample. What it means is you take a set of samples, and 10 samples, let's say, but one time you do the measurement, and if it is greater than certain defect, D is for defect. certain defect measure. If it's greater, you reject right away. Okay? That is a single sampling. You took 10 samples, you measured the defect, whatever it is, or the quality parameter. If it exceeds a certain standard, then you reject the lot. That's it, single sampling. The double sampling, that happens if the value is between an accepted amount and a rejected amount. If it is between these values, a value where you accept and a value where you reject, so A for accept, R for reject. If it is between, then you have to do a second set of sampling. That means not you take one more sample, you take another set of 10 samples from different locations, and then again, you would determine the acceptance level and the rejection level. So again, I don't expect you, this is just letting you know, but I won't ask anything like that in the exam. But you want to know and be aware of it when you're working environment, you want to know these things, but you know where to find them. You open a resource book and you know where to find them. Multiple or sequential sampling. So in this case, you have a number of defects, and then you have number of samples here. If you take 25, or let's say 30 samples, and you found less than one defect, you accept. This is for a particular quality. They're not all the same, right? There's not the same graph for every quality parameter you measure. We're talking about X random parameter, fat content, or presence of metal, or whatever it is. So it would be a different graph for a different parameter. And if you took, let's say, 10 samples and you found 7 defects, or 7 defective samples, you reject. Okay? And if it's in between, you continue sampling until you make a decision to either reject or accept. Again, this is FYI, so that if you see it in the future, you understand it, but you have resources to go back to. You will not remember, even if you remember it for the quiz, you're not going to remember it when you start working. So these are information for you to know where to find them and to understand them. Okay, when monitoring food safety by measurement of mycotoxins, okay, so this is toxin. Many randomly selected samples are required. A single sampling plan is required. Sampling error is many times greater than analytical error. Abi? A is correct, but what do you say, Shri? A and C, yes. So C is very important in this case, because if you do a sampling mistake and then you found that, oh, I don't have aflatoxin, and you accept it, but in fact you do have it, that's a greater risk on the consumer. So in this case, sampling error is many times greater than analytical error, because usually when you measure aflatoxin, you're probably doing an HPLC-MS method. The analytical error there is not as huge, but if you make a sampling error, the decision is more consequential than, not consequential. It's more of value, it's more of an impact, more impact. Okay. So that brings me to consumer risk. You want to make sure that the probability that you have a wrong answer or a wrong decision is less than 5% for consumer. For vendor, there is a risk because of rejecting something that is really valuable, so money. So here, it is still higher. Your risk can be up to 5 to 10% of the lots for rejecting a good quality population. And here, for accepting a poor quality population, the risk should be less than 5%. Abby, I saw your hand up. Previous slide. Oh, well, it would be if you're measuring, usually there is no such situation. So here I'm emphasizing the sampling error, right? Even if it is analysis of ash volume, right? If you do the sampling wrong, it's going to impact the decision. But in this particular case, sampling error is a much bigger risk than your error from analysis. What's that? Proven?
Yeah.
Yeah, you want to do your analysis right, but the consequence of wrong sampling is greater than the consequence of analysis. The consequence, if you sample wrong and you do your analysis correct, you're going to get into a big trouble. Usually analysis won't have that much of a risk. If you're well trained and if you're working in a lab that has trained you well on the assay, and that's usually the case, your error is not as huge as when you have a sampling. Yeah, because they're not going to bring any random person to do the analysis. They're trained to do the analysis. So a sampling plan should be simple and flexible, protect consumers and vendors, and ensure reliability of results. So this you should know. Okay, that's simple. You need to keep this in mind, but then how to construct a sampling plan, that will come later, and you know where to find the resources. Here's an example of sampling from flour. It's a homogeneous product. It's already been milled, and there is no like protein content, for example, should be normally distributed in the flour sample. Okay, so if I have 100 sacks of flour samples that I produced in one day, and the flour sacks or bags are about 40 pounds or 100 pounds, doesn't matter. But let's say I have 100. So I won't take every bag and sample from it. I would take the square root of the number of sacks in a lot. So if I have 100, the square root of 100 is 10. Okay, so I would want to take 10 bags randomly though. And I'll talk about how we do random. So you know the random number tables. So you would number your bags and you have a random number table that contain the numbers of your bag, and then close your eyes and select the number. Okay, so you do that 10 times and you get 10 bags. And when you get the 10 bags, obviously you have 40 pounds, 100 pounds. You're not going to measure all the flour in there. So you want to take a sample from each of these 10 bags. So the way you do it in there, that's an official method of sampling of flour. So you draw a sample from a corner, diagonal to the center, and then you draw a sample from the other corner, also diagonal, opposite corner, diagonal to the center. Then the samples you have are stored in clean, dry, well-sealed container. So now you have your samples from each bag to be analyzed. So again, if you have a homogeneous sample, already a flour that's already homogeneous and milled for you, or milk, let's say, already homogeneous product, sampling can be taken from any location. If you have a heterogeneous sample, you take from multiple locations or you homogenize your sample. So either you take eat the whole thing and mill it, but let's say grind it. Or if it's a lot, you can do grinding everything or homogenizing everything. You just take samples from different multiple locations so that the sampling or the samples are representative of the lot. There is manual sampling and there's mechanical sampling. So mechanical sampling is less biased, so that would be great to have. If you are analyzing samples online, you can have mechanical sampling online timed. Every 10 minutes, a sample is pulled out mechanically without any human bias. Or there's manual sampling, which is based on human error. So I will talk briefly about statistical consideration when you have sampling. Sometimes you cannot have statistical consideration. We call it non-probability sampling. And here where you have either really a truck full of grain, you can't do anything here statistically with that. It has to be with judgment. Okay, the guy knows what he's doing. He's going to select samples and give it to the lab. Because it just, you cannot do statistics here. No numbering, no picking random. Or restricted sampling. You have a silo of grains or whatever it is, a big container. You cannot reach to the bottom. So you have to take from the top, whatever you can reach to take sample and analyze. So it's not the perfect situation in this case, but there is not much you can do about it. But oftentimes you can do probability sampling. That's when you have your product or whatever it is, ingredient or product in individual containers where you have either cans or bags. So you can do probability testing or sampling. The random sampling is what I just talked about. You number your cans or you number your boxes or your bags, random numbers, and have a random number table and select your samples randomly based on these numbers. Systematic sampling happens usually with the mechanical sampling. That's basically sampling, let's say, of milk online. Every 10 minutes, you draw a sample, and that would be systematic sampling. That means every time point, you pull a sample mechanically and test. Stratified and cluster sampling, these are very, very similar. So let's say your company produces soup or tomato paste, and they have multiple location plants. So what you do is you stratify. So you give every plant a number, whatever it is, and then you would select samples from each plant randomly by numbering, and then you test them. The cluster sampling is the same thing. You have multiple plants, you number them, but instead of taking sample from every plant, you randomly select plants for testing. So it really depends on how big the company is, how big the lot is, and what's the purpose of the testing. And because also always remember that there's cost associated with sampling. How many samples you pull, how much analysis you do, all cost money.
With cluster, then create like more risk.
What's that?
With cluster sampling, create more risk.
Well, again, it depends on the purpose. Sometimes if the purpose is having to do with consumer risk, they might not do this kind of sampling, they might do the stratified. So it depends on the purpose. If you're just doing a general quality check or general nutrient, you can do that. So it depends and ask yourself, what's the purpose of my analysis or sampling? And then you choose one. Yeah. Oh, composite sampling. That's the example I gave you earlier of the FDA and FSIS. So that means you take random samples by random sampling, and then you combine them to produce a composite. And that's exactly what you do for nutrition labeling. Let's say you take 12 random samples from the lot, combine them, open the cans, open the box, whatever it is, combine them, homogenize them, and then from there you take 6 subsamples for testing. So that's what composite sampling is. The following are potential problems associated with sampling that may result in reduced reliability. Non-statistically viable convenience, poor storage conditions, mislabeling of samples. Michael? That's a good answer. Yeah. It's all of the above. Yes. So, sometimes there's no choice and you have to do that, but there's a risk associated with it. Poor storage conditions going to change the characteristics of your product. Mislabeling. Oh, that's a big blunder. If you don't want to do that, yes, Sandy. When Amy, this situation. You can do statistics. You cannot number. You just have you have to take sample from a truck. Go figure, right? Yeah, we went over that. Okay. Reduction of particle size. This is really important. One time, okay, this might be relevant to you for capstone. So I walk into the lab, there's a capstone group doing mojo or mojiner or mojonine, or you want to say it, you can say it. It's a fat assay, fat analysis. And they have, their product was potato chips. And you'll see later we have specific flasks for mojiner to extract the fat and measure the fat. So I walk in and they're shaking, the solvent of the sample. I look inside, I find the potato chips as is. It's like, what are you doing? Extracting the fat. You should grind your sample first. You have to increase the surface area. They didn't know or they forgot from food analysis. So you remember when you're doing your capstone, if you have a non-homogeneous sample or big chunk of the sample, it has to be ground into a 20 mesh or a 40 mesh, depending on your assay. Okay, and if you remember what is a mesh size, what does it mean, a 20 mesh?
The amount of holes for one linear area.
Inch. Yes, you were close. So 20 openings in 1 linear inch. So the more openings you have, the smaller is your particle size. And for extraction experiments, you want the smaller size. So you enhance your surface area for extraction. So, okay, another capstone thing, let's say they are doing some sort of a bar and it has high moisture. And how do you grind something that is sticky with high moisture? It's not going to be easy. One thing you would do, you freeze it. So you grind it when it's frozen, call that cryogrind. So in this case, you are able to grind and not lose moisture or fat from your sample when you're grinding a high fat, high moisture sample. So that is, and here in this picture is just showing you like a grinder with different sieves, where you can determine the mesh size by putting different sieves. So your particles that pass through certain mesh size, oh, they're below 20 mesh, they pass through 20 mesh. Or they're below 40 mesh, they pass through 40 mesh sieves. Okay, enzyme elimination or control. Why do we care? We talked about it maybe briefly last time. And how do we control it? Let's first answer, why do we care? Why do we have to limit enzyme reaction when we're preparing the sample and storing it until we run the analysis? Can you give me an example where enzyme potentially can change something, Kaya?
If there's too much enzyme activity, can't it make it go bad?
It can, yes. Can anybody give an example of an enzyme? Go ahead.
I was thinking if you're cutting up a piece of fruit
Yeah.
Like a banana, like the enzymes interact with the air and.
Yeah,
change the
polyphenol
result.
oxidase enzyme, absolutely. So that is one. Another enzyme we gave an example about is lipase lipoxygenase, oxidation. So especially, this is really important if we are actually measuring lipid oxidation. So as the product is, you don't want to measure lipid oxidation during the storage of a prepared sample. So you don't want your lipoxygenase to work. So if you're measuring, let's see, lactose content. You don't want any lactase enzyme in there that would hydrolyze your lactase to give you glucose and galactose. So you don't want any change in the chemical composition of your product. Or if you're doing sensory analysis and then you have enzymes that break your protein and break your fat, your whole sensory perception will be different. So we do not want enzymes to act. How can we prevent that? Yes, Elisa? You can treat it, yeah, with you can denature the enzyme, but sometimes you don't want to heat your sample for other particular reasons.
Depending on what the thing you're testing, you use acid,
pH, you can change the pH if it's not going to impact anything else you're testing. And one last very simple measure, very simple. If you don't want to change anything, you don't want to heat, you don't want to change pH. Go ahead, Lynn. Freeze it. Thank you. Yes. So oftentimes we store under -20, sometimes -40. We prepare the sample. We take every precautions to seal it. Sometimes we remove the oxygen by flushing with nitrogen, seal well. If we don't want light, we cover it with aluminum foil and put it in the freezer. Yes, true.
Could you also add like enzyme? Could you add something to block the receptor?
Okay, so you can add inhibitor. Enzyme inhibitors is what you're looking for. You can do that as well. But again, you have to think of cost. No problem. It happens with everybody. Okay. Avoid lipid oxidation. We said that, and we kind of talked about, okay, flushing with nitrogen, away from light, making sure lipoxygenase is not working. What else we can do to prevent oxidation? There's another simple measure. Yes, Shinu? Extract the lipid out. You can do that, yes, but if you're doing sensory analysis, say you don't want to extract the fat, you want to keep it as is. What you can do sometimes is just put antioxidants in there. Of course, we want to limit microbial growth or contamination because when microbes grow, there's a lot of things that happen. They also secrete enzymes, and the enzymes can ferment the food, change the composition of the food, spoil whatever you are storing for analysis. So you don't want that. What can you do? You can add preservatives or store for storage under refrigerated or frozen conditions. Okay, and well sealed is very important because you don't want microbes to go in and spoil your food. Some microbes grow under refrigerated conditions as well. So whatever you do, you want to avoid sample alteration. You do not want to change the sample, so you have to always think, okay, what is the analysis I'm after, and how do I protect my sample until I'm ready to analyze? Sometimes I can give you an example. We have one of my students in the lab, they're doing a storage study. And basically they're storing milled flour from particular grain. And then they have to take a sample after certain storage time, 1 month, 2 months, 3 months, 6 months, 8 months. But there are assays that are exhaustive. Let's say she wants to determine carotenoid or hydroxycinemic acid. She doesn't want to do that on 3 samples every time she pulls out a sample. She wants to do the assay on all the samples to save time. So what she does, she pulls out the sample, put them in out of the storage, put them in sealed container or flush with nitrogen, put in sealed containers, cover with aluminum foil in the freezer until she has all of her samples by the 9 months of storage, and then she can do the analysis on all of them. So whatever she needs to do, she makes sure that she doesn't alter the sample until time of analysis. So that's very important. Okay. Finally, you guys, this is study question 7 from your book. For extra credit, +5, not +2, +5. So I want you to think about it, type up your answer or write down your answer. I don't care, bring it next Monday if you want. Just bring it here for extra 5 points. Okay, there's 5 minutes. Do I start a new topic or do I let you go? Who wants to stay? Nobody wants to stay? Come on, it's one of my favorite topics. Okay, off you go. We'll see you on Wednesday.
We do them
Okay.
Yeah. Most of my classes are still doing them online, but I forgot. Yeah.
Yeah, let me stop the recording.